Greetings everyone and welcome to another Genetic Engineering and Biotechnology News webinar. Our presentation today is entitled Multiplexing PCR Design Made Easy. I'm Jeff Bogaliskas, Technical Editor for GEN, and I'll be the moderator for today's webinar presentation. Walk into any molecular biology lab these days and on any given day, invariably there'll be a PCR reaction ongoing. While PCR has been put to good use over the past 40 years and its ubiquitous nature makes it indispensable towards daily laboratory operations, the technique is anything but mundane, as researchers and companies are always finding new ways to improve the reaction. Recently, investigators have begun to capitalize on the fundamental nature of PCR through multiplexing reactions, saving them valuable time and money. Though multiplexing reactions can be a benefit to lab productivity, they are not without their drawbacks. The added parameters of multiplexed PCR reactions can dramatically increase the complexity of experimental design and validation to frustrating levels for many researchers. Thankfully, a host of computer software has been created to ease molecular biologists' multiplexing design woes. Let's meet our speakers for this webinar who will tell us how PanelPlex software has allowed them to quickly and efficiently design panels for various disease identification and molecular diagnostic purposes. Arjang Hasibi is the founder and CEO of InSilixa, headquartered in Sunnyvale, California. Dr. Hasibi will tell us how PanelPlex has helped InSilixa develop a screening platform for upper respiratory pathogens. Aditya Rajagopal is founder and CSO of ChromaCode, located in Carlsbad, California. Dr. Rajagopal will tell us how PanelPlex was critical in the development of a multiviral molecular diagnostic tool. Finally, John Santalucia is the founder and CEO of DNA Software, located in beautiful Ann Arbor, Michigan. Dr. Santalucia will tell us about some of the philosophy, science, and pitfalls behind PCR primer design and how PanelPlex has simplified the process. Before our speakers get started, I want to encourage the audience to submit questions for our Q&A session at the end of the presentations. We'll try to answer as many questions as we can, so simply type your question into the box below the presentation screen and hit submit. Okay, with that, let's begin our webinar and hear what our speakers have to say. Arjang, you have our attention.
Thank you, Jeff, for the introduction, and hello, everyone. My name is Arjang Hasibi, and I'm CEO and founder of InSilixa, a startup company in the area of molecular diagnostic here in Silicon Valley, California. Our company has been working with DNA Software, John, and the rest of their team members for approximately right now 3 years. And we have been using their tools extensively, almost all of their tools, both in our R&D projects and also for the product development. I guess that is why they have kindly invited me to this webinar to share my experience and show you some of the results that we have got with their tools. Specifically, with the tools that are relevant to the PCR primer design, like PanelPlex and etc. We have been using DNA software tools for design of many different panels. However, in this presentation, I will focus on showing you the results from a relatively large, panel that had detection, genotyping, and also quantification for respiratory infections. First, let's discuss quickly what we're trying to build at InSilixa and why is it relevant to the tools that DNA Software has been developing. Here I'm showing you one of our products. It's called the Hydra platform, which is a sample to answer nucleic acid amplification testing system. As shown over here, a sample from a patient is taken, put into a cartridge, a disposable cartridge, and this cartridge is put into a random access instrument, and in about 90 minutes you can get the results of the test. And in this, in the Hydra platform, we can do a very high multiplex nucleic acid amplification testing. We can quantify many different sequences. We can genotype, we can do all of them, and the level of multiplexing can go up to 1024, and it's all enabled by the CMOS biochip that is embedded in the cartridge, which does not only the heating and cooling, but also genotyping and so forth. What is important is actually the method that we use in the cartridges. And the method that we have is shown over here, and I'm going to spend a few minutes on it, and you will see how does it relate to PanelPlex. It's called the solid phase multiplex quantitative RT-PCR, which also includes melt analysis in all-in-one reaction chamber. So it's a closed tube or homogeneous assay. If you look at the left of the slide, you will see what essentially we have in the cartridges. You know, when the total nucleic acid is extracted, essentially what you have is a pool of DNA and RNA that you're trying to see if specific regions of interest are present to identify organisms. And in many cases, those regions of interest that you have might be, you know, location within the genome of the organisms that there are clinically relevant mutations that you wanted to detect. However, when we have this in the reaction chamber, we also put in the reaction chamber an oligonucleotide array with capture probes with unique sequences that we have. And these are, you know, basically embedded on a sensor array which can detect the capturing of the nucleic acid or the hybridization of the target with the nucleic acid in real time when the solution is present. So if you look at the middle, essentially what the system does is as follows. When I have the pool of the microbial nucleic acid and all of them in the sample, we perform multiplex nucleic acid to amplify the regions of interest which are over there. So there are many forward and reverse primers over there, you know, up to 50 or 100. They might be, you know, concurrently present. We amplify all those regions of interest. We amplify them asymmetrically. That means the concentration of the forward is much more on the reverse primers to create, you know, single-stranded, more single-stranded after the plateau of PCR. So it's an asymmetric PCR. And after this is done in the reaction, we basically, the probes that you have on the surface can capture the single strands. And we have 1,024 probe locations that we can, you know, populate. They can, you know, essentially probe the amplicons, and you know, you can check if there's very specific sequences are present or not. We not only do the capturing, but also we can do the solid phase melt. That means that when the, you know, sort of targets are hybridized, we can increase the temperature after that and see at what temperature, at what point they detach. And basically by doing this, we can better identify or differentiate between the sequences and do single nucleotide polymorphism and other things over there. The whole system is closed tube. There's one reaction chamber. There's no washing. Nothing is necessary. And the paper which describes this is going to come pretty soon in Nature Biotechnologies, accepted, you know, in a month or so. This will come out. The panel that we intended to actually implement on Hydra, one of them was this comprehensive upper respiratory panel or URP panel. It's a fairly large one. It has 27 viruses, 3 bacteria panels, 2 controls, including human host control, to check if the sample is actually taken correctly. As you can see in this list, there are RNA viruses, there are DNA viruses, there are bacteria, and all sorts of things. One important thing is, you know, a lot of them are essentially detection. You just have to know their presence or not. But some of them, like the flu antiviral resistance, is basically a genotyping platform. You know, you have mutations across the different segments of the organism that you have to check, and if they're present, then you know that, you know, this strain, for example, is Tamiflu resistant and everything. One important thing is, you know, beside the size of it, you know, we had to deal with a fairly large database that we downloaded from GenBank. And as you can see on the number of strains that we had that, you know, we had to detect to satisfy the inclusivity and so forth, the numbers were in, you know, tens of thousands, you know, 12,000. But in some of the organisms like the, you know, bacteria, because they were less variant, they were smaller over there. But we had to also include all of them in the design that we have. Although our panel, our biochip and the Hydra system was highly multiplex, you know, dealing with this inclusivity and exclusivity and designing primers which are that general was a true channel without interfering with each other. Okay, here's what actually we put together, which we call it our assay development pipeline. It has 3 components. Going from left, first we have to set up, you know, in order to design the assay, and the tasks are pretty straightforward. First, you have to know what exactly you wanted to detect, you know, from a genomic point of view. So that's the assay specification, requirements, you know, inclusivity, exclusivity clearly. You have to know all those things based on the strain. You have to create a sequence database That can be cumbersome. There's a lot of repeated sequences. There's a lot of issues with what you download from NCBI and all of them. But essentially, the setup is before actually use any of the tools like the PanelPlex you have to do. But then you go actually to the bioinformatics part, which in our case was automated primer design, mainly done by PanelPlex. We had rigorous review that we did using the other tools of the DNA software. We did the probe design as well. Remember, in our system, we had surface probes or solid phase melt probes that we had to design to, probe the amplicons. And also we have, when we get to the, multiplex PCR, we had to do prediction, we had to do debugging, and all of them fall into the bioinformatic part of it. The experimental part of the work, as far as our pipeline, work hand in hand with the bioinformatic part. Initially, if you look at the right side of it, initially we did the single place primer qualification. We generally get a lot of primer candidates, and we had to screen them. We ranked them and down select them to the ones that empirically we liked. Then after that, we characterized them rigorously to the nth degree. We had to check them for variability, tolerance to variability of the sequence and the template. We had to check the asymmetric PCR efficiency, the yields that they had, and also the PCR efficiency. And also did the guard band testing, basically changing the temperature and salt and all of those things, and see if the sort of panel is actually robust. And we used the DNA software tools to predict, and then validate it with the tools that we have. And we did also the probe testing. And after the primers were qualified, we obviously did the multiplex testing and assembly and debugging them. And after all of these things were done and all the requirements were satisfied, that was essentially when we called the project was successful. Now here is what we did and why our partnership with DNA software mattered. We enabled all of our bioinformatic pipeline by using the tools that DNA software had, except some of the other tools like the normal BLAST and things like that. The automated primer design was done by PanelPlex. The reviews were mainly done by ThermoBLAST and OmpDE. Essentially, OmpDE is it was the bread and butter of that part of it. For the probe design, we not only use the ThermoBLAST and OmpDE, but we also use another tool that I don't know if you guys are familiar with. It's the oligokinetic simulator, because we have to look at the hybridization kinetic and some of those things. And when we were trying to debug and check the multiplex PCR sort of assembly, we also used the ThermoBLAST and OmpDE. And there was one tool that went beyond all of these things, and that was VisualOmp. It's one of my own favorite tools, because to communicate between the scientists and between the bioinformatic group and the other ones, I mean, you just need a tool that anybody can use and they can have on their desktop and everything, and that was VisualOmp. So that's how we essentially put everything together. So here are the results, or the highlights. The first one of highlights was whatever we got out of PanelPlex. If you put the right inputs in it, were awesome primers. If you get anything out of it, that means that if your constraints were feasible, you would get extremely good panels. And we would get, between 4 to 5 at least set of primers for each of the panels in the first run. And when we tested them, about 75% of them, I actually checked this number before this presentation, 75% of the primers generated by PanelPlex meet the requirement in the first run, which was extremely high. That means they were working, and then it was sometimes hard to decide which one do you want to keep, which one you don't want to keep and not. And this is an example of the results that we get out of the screening. For example, we did a dilution series, and this is, I think, adenovirus B. We call it ADV3. I think the acronym was that. And this is a dilution series that we had the top figure of it. As you can see, the green parts are for each dilution, 1 to 10, and the red is the NTC. So the NTC, and there's human background in the NTC as well. So it's not just pure water that you have. So it looked good. And when we did the efficiency calculation, you could see that the efficiency was around 0.995 across that, pretty linear. And this was a typical results that we get out of these primers, which are extremely encouraging if you know what you're dealing with. Another important highlight was when we put everything together. So obviously, computationally, initially we checked that there's no primer dimer, and none of those things happen, and that was the easy part of it. When you generally put the multiplex together, some of the panels, they would not get annihilated, but the efficiency would change a little bit. Some of them, 10%, 15%, yields will go down and everything. So you would lose, the pristine singleplex quality of it. And then you go to the level that you try to see, okay, what's going on? Are there any interaction or not? The common interactions that people, consider like primer dimer or amplicon primer interactions and everything, those are easily in the tools you can find out. But there are another sets of them that will come out. And here are, for example, two examples that we see. So when we put the panel together, we saw that the H1N1 panel was somehow in the presence of adenovirus E panel, the efficiency would go down. And we look at it, there was no primer dimer, there was nothing that would happen. And then when we went deeper and we look at with ThermoBLAST and Opti, we went deeper, we found out the situation like this. In this situation, this shows the duplex of the two primers within these two panels. One of them, the top one, is the excess primer for adeno E, which is 300 nanomolar. And the other one is a limiting primer H1N1, which is about 50 nanomolar. Remember, this is asymmetric PCR. The likelihood of this happening in the PCR temperatures is extremely low. This is about, I think, 0.01 or some ridiculous low percentage of happening. And the TM is about 28 degree, delta G is minus 2.9 kilocalorie per mole. But if you look at it carefully, you will see that this is not an exponential growth, but there is a chance that the limiting primer might extend and get essentially, deactivated, because then, it cannot work with the right template and everything. And we associated this with that reduction in the yield of H1N1, and we quickly fixed it by, adding, I don't remember what we did. We added a, intentional mutation or, choose another primer that we have. In example 2 below that, one of our, PCR panels had a lower yield, and there was no reason for it. It was not exactly 100% and everything. And when we look at the structure, and this is the enterovirus D68 panel, you would see that 23% of the limiting primers were in this kind of hairpin structure, and you can easily see that, and you can increase the concentration to the level that you want, and everything would actually go well. So these things are extremely important when you want to polish your multiplex, and these were all enabled with what we did with the DNA software tools. The third highlight, which is essentially my conclusion based on this project or this partnership that we had, was our development time was really fast. And the main reason was the amount of redo that we had to do on the primer design was just did not exist. It was just most of our problem was which primer to use and which one would play better. So the iterations were very limited, the number of iterations limited. So we quickly finished the project, and essentially there was the number of full-time people that we need on this one, especially on the experimental part, was extremely low. So we did this whole project in about 4 months with 3 people, which is unheard of. So in conclusion, what I want to say, why essentially we use DNA software tools, and this is completely from a technical point of view. Maybe about 10 years ago, if you asked me that can you have a fully automated multiplex primer design pipeline, I would say, yeah, probably not. But right now it's possible. And after going through this project, I would see the models from DNA software, thermodynamic models are excellent. You can actually create real robust assays. And you can do the gardening, sort of in a way that, no matter what, the panel will work. The other thing is the tools that the DNA software has. I mean, we use them every day. You don't need to be for all of them. You don't need to be a sophisticated bioinformatician. I mean, specifically Visual Amp and all of them is just a must-have. So they have been a right partner for us. I don't know for others, and their technical expertise is extremely good. They have been really helpful, in the design, and this is not just on the validation, but also on making the methods that we have much better. So the result was, we were worried about how much the design of these panels would take time when we're actually pushing our products, and we are on sort of limited budget in a startup. And the result was, yeah, we didn't need to worry about it that much. It was just straightforward. It was a lot of work, but it was straightforward. And we didn't need to do, again, mindless coding around Primer 3 or deal with any of those freewares that actually they're okay, but they're not going to get you into a robust system. Thank you.
Thanks very much, Arjang. That was a really great presentation to start off this webinar. Before we move on to the next presentation, I want to remind the audience to submit questions for our Q&A session at the end of the talks. We'll try to get to as many questions as we can, so simply type your question into the box below the presentation screen and hit submit. All right, with that out of the way, Aditya, the floor is yours.
Jeff, thank you for the introduction. Joe and John, thank you for having me join you for this webinar. I'm excited to talk to you about what we've been up to at ChromaCode. We're enabling mid-plex diagnostics on existing qPCR instruments. As you guys know, PCR was a seminal discovery. Kerry Mullis came up with the concept while on a memorable road trip up in Northern California. And it's fundamentally changed the way we perform microbiology. In fact, PCR has conquered the world because it's fast, cheap, and ubiquitous. In traditional qPCR instruments and assays, you can measure 4 to 6 things per reaction, 1 per color channel. Although the instrumentation is ubiquitous, it is critically limited in the number of things it can measure at once. At ChromaCode, we're a bunch of biochemists, engineers, and machine learning experts, and we've reimagined multiplexing in existing qPCR instruments. With high-definition PCR, we can enable you to run a panel of tests in a single PCR reaction. Markers for oncology, for infectious diseases, for gene expression. We've designed a set of multiplexed chemistries that allow us to engineer PCR curves. Now with that same PCR instrument, we can read out many targets in each detection channel. And with our cloud software, we can interpret these curves to map back to combinations of targets that were present in a reaction. One of the aspects we engineer is the color intensity of each curve. With HDPCR, each target that's measured in a single color channel has a unique curve intensity. For example, if target A is present, it generates a curve with a brightness of 50. If target B is present, it generates a curve with a brightness of 100. And if target C is present, it generates a curve with a brightness of 200. However, every combination of targets also has a unique brightness. Let's take another look at how this works. Consider a reaction where target A is present. In this single color channel, target A's presence generates a PCR curve with a brightness of 50. If instead target B were present in the reaction, the curve would have an endpoint intensity of 100. Target C similarly would have a signal intensity of 200. Indeed, each one of these targets, if they were present alone, would have a unique curve intensity. But with HDPCR, each combination of targets also has a unique curve intensity. So for example, if we have a reaction that has both targets A and B present, the resulting curve would be the addition of the curve for target A alone and the curve for target B alone. With HDPCR, we can fit many targets into a single qPCR curve. In fact, it is the only method that allows us to map a set of targets to a unique curve signature. We call these curve signatures levels, and you can see here how the curves themselves fall cleanly into the level bands. We've thought a lot about chemical interactions in the PCR itself, and we've engineered the qPCR chemistry and developed mathematics and signal processing to drive uniform performance within an instrument. Here you can see a number of qPCR assays that have been run by different operators on different days on a same instrument. This consistent intra-instrument performance drives consistent inter-instrument performance. The same assay performs reproducibly and robustly across a number of qPCR instruments. Now I'd like to take you through a few examples using our respiratory viral panel platform demonstrator assay. The graph that you see in the background is a screenshot from our cloud software. In this particular reaction, we had 3 targets that were present that were detected in a single color channel. We had 10,000 copies of RSV-A, 10,000 copies of human rhinovirus, and 10,000 copies of RSV-B. Had the reaction only had RSV-A, the qPCR curve would have terminated in the band that you see highlighted in black. If instead there had only been human rhinovirus, the curve would have terminated in the band that you see highlighted here in blue. Similarly, if RSV-B had only been present, you would have had a curve that would have terminated in the band you see highlighted in red. But because all 3 targets were present, all 3 sets of primers and probes were amplified in this single color channel, resulting in a curve that uniquely terminated in the band highlighted in green. HDPCR works the same way with clinical samples. In this particular sample, influenza A H3 was present. In our assay, we have primer and probe pairs for both the pan-influenza A marker and the H3 subtype. Because both marker sets were amplified, the resulting curve from this clinical sample terminated in the band highlighted in green. HDPCR is engineered so that curves terminate in deterministic bands, regardless of the concentration of the targets that are present in the reaction. This is ChromaCode HDPCR. It's a controllable chemistry engineered for robust performance across targets and concentrations. It's mathematics and algorithms to interpret curve signatures and deliver consistent results on multiple platforms. All of this enables mid-plexing on qPCR instruments. But wait, there's one more thing. We can not only enhance qPCR instrumentation, but we can also enhance digital PCR instrumentation. HDPCR lets you take a traditional 2-color digital PCR instrument platform and measure 10 to 20 targets per reaction. This is only possible because we're able to model the complex interactions that take place in multiplexed PCR. And that's where DNA software comes in. John and his team have done a lot of hard work to build a tool, Visual OMP, that's very insightful and helpful in our multiplexed assay design. In fact, we're currently evaluating their PanelPlex, which is the next evolution in InSilixa design software. We're looking forward to working with DNA software in the upcoming months. To summarize, we are ChromaCode. We're bringing mid-plex panels to a PCR instrument near you. Thank you for your time today, and John and Joe, thank you for hosting me for this webinar. Please feel free to reach out to us if you have any questions.
Thanks, Aditya. That was a very detailed presentation. Before we move on to our final presentation, I want to remind the audience once again to submit questions for a Q&A session after the last talk. We'll try to get to as many questions as we can, so simply type your question into the Q&A box below the presentation screen and hit submit. All right, let's move on to our final presentation. John, the audience is listening.
All right, so I mentioned that I'll be your presenter for the third part of this webinar. I'm John Sinalochia, the founder and CEO of DNA Software. The DNA Software was founded to commercialize research done in my lab and to extend upon that research and go further to deliver software for automated designs. We've been in business for more than 17 years, and we've helped numerous organizations with their molecular diagnostics. We pride ourselves in particular with solving some of the most challenging problems in oligonucleotide design. And we also have used the knowledge of doing previous projects to distill it down into software that is very easy to use. All right, so today I'll be telling you about the challenges of multiplex PCR and our unique approach to that problem. All right, so let's first talk about this. PCR is a widely used method and has been engineered in numerous parts of its components. But PCR, one aspect of PCR that has been neglected is primer design. If you think about instrumentation, like the instrumentation offered by InSilixa, for example, many companies have worked very hard to miniaturize PCR, to introduce microfluidics, to improve the sample preparation techniques with kits and robotics, microfluidics. They've done a lot with improving the master mixes that go in there, the buffers and additives, and they've done a lot with the data analysis. But one part of PCR that has been largely empirical has been the design of primers. And this is despite the fact that PCR, if you think about it, is a relatively simple reaction. You have your genomic DNA, you may have some contamination in there, but you really only have a polymerase and some salts. And that is something that should be subject to scientific investigation. And we've done exactly that at DNA Software to try to understand the causes of failures of PCR. Now, many researchers, not knowing how to design PCR, will put forth the effort to combine various free software into a PCR design pipeline, which is shown here. And we'll be showing that each one of the stages that typical design pipeline like this have difficulties or problems with the way they're set up. One of the first problems is this funnel that I've shown here. If you look at the funnel, the idea here is that computation is limited, and so we should take a wide number of primer candidates and then as quickly as possible filter those down to a few candidates because the computational methods get more and more expensive. That very philosophy of computation being scarce is something that leads to problems with not finding the proper primers. So for example, some of the problems with multiple sequence alignment, which is often used to find conserved regions of a genome, is that the sequences, what we really care about for PCR is not sequence identity conservation, but we care about how well will a primer bind to different members of a group of sequences. And multiple sequence alignment really does not give that. Another aspect that folks will do is they'll use a free software like Primer3, which does indeed do a wonderful job predicting two-state melting temperatures and predicting primer dimers even. But it is neglecting the competing folding. So they might use a separate program like mFold to predict the secondary structure of DNA, but they don't combine those two to understand the competition. And lastly, they may use a program like BLAST to check the specificity of their primers and check to see if their primers, for example, bind to anything in the human genome. But we'll see again that BLAST is really not the correct tool to use for that problem. And lastly, this design pipeline is really kind of designed around a single target at a time. And multiplex PCR is quite different than that. You have many targets in the same reaction, and you need to account for the system effects. All right, so because the current approaches for designing PCR don't work that well, researchers are left with the empirical optimization approach. And typically the idea here would be to start by optimizing individual singleplexes and getting those to work. And certainly if a PCR doesn't work in a singleplex, it's not going to work or get better when you make it into a multiplex. But then the idea is to try to combine those singleplexes into larger and larger groups And when a single plex doesn't work or interferes with one of the other members of the multiplex, to fix that single plex. However, the process of fixing that single plex causes other reactions to fail, and it's not known why those other reactions to fail. And so it typically takes a PhD level scientist 3 to 6 months to get a 10-plex PCR to work, if at all. And this results in a sort of, whack-a-mole situation where you might have a 7-plex working, you go to make it an 8-plex, and boom, a different primer doesn't work. And then you have to try to re-optimize and go to scratch often. This is sort of a whack-a-mole situation where different problems keep popping up. And talking about the multiplex problem, the problem here is to try to get sets of primers that are mutually compatible, where compatible means that the primer sets amplify with similar efficiency, don't cross-hybridize, and don't form false amplicons. Let's take, for example, a 30-plex, much like the size of the reaction we did for InSilixa. So if you were to try to keep, let's say in a 30-plex, you keep 10 primer pairs candidates for each of the targets, each of the 30 targets. So to start with, you've got 300 forward primer candidates and 300 reverse primer candidates plus probes. All right, and you might ask, well, how many possible combinations are there? Well, for each panel, there's 10 combinations of primers. So as you add panels, it multiplies 10 times 10 times 10. In the case of a 30-plex, it'd be 10 to the 30th power possible multiplex solutions, a number that is 10 million times larger than the Avogadro's number. And as you go to even larger plexes, consider like a 100-plex, 10 to the 100 power would be a truly astronomical number, larger than the number of particles in the universe. So we have a problem here. We have an exponential explosion, and each trial multiplex combination requires an extensive amount of computation for each one to check the specificity, for example, and also to check for cross-reactivity and false amplicons and competing folding. So with such a huge space of possible multiplexes, there's a need for a different approach. So our goal at DNA Software is to solve the multiplex PCR design problem outright. And our approach to solving the PCR design problem, is three-pronged. We look at the science of the problem. We investigate the causes of failures of PCR and implement those understandings into our software. Further, we develop new algorithms for predicting the secondary structure that competes with primer hybridization, for calculating off-target mishybridization reactions that lead to false positives and false negatives, as we'll see, and also accounting for this multiplexing effect I talked about in the last slide. And lastly, we bring to bear massive cloud computing resources so that we can address the full challenge of the problem, including modern large sequence databases. All right, so I'm going to be giving you a brief overview of the 4 most common problems in multiplex design. This is what I'm going to be giving you today is an abbreviated version of a larger discussion that we've presented in 2 free white papers and a webinar that you can view at a different time. They're available on our website at DNA Software or on YouTube. But I'll give a brief introduction to those today, just to sort of set the stage. So multiplex panel design challenges include the following. The first challenge is that targets need to be amplified at the same rate. But often in real multiplex PCR reactions, one of the amplicons can take over the reaction, resulting in amplification at uneven rates. And we'll talk about what the causes of that are. Another artifact that can occur in multiplex PCR is due to cross hybridization, where a primer from one target binds to the amplicon of a different target. Or you can get background amplification, where primers amplify some contaminating DNA that's present in the reaction. And we'll see that both of these problems are not addressed with using a blast search. In addition, there is the need in the 21st century to include large sequence databases as a part of your design, and in particular, the design of inclusivity, exclusivity, and a background list of genomes. And then lastly is the issue of getting everything to work well together in a multiplex PCR, taking into account the fact that multiplex PCR is a complex system with many interacting variables. All right, so one of the first effects that we would like to discuss today is the issue of target secondary structure. On the left-hand side is the model that is most widely used for predicting the thermodynamics, the melting temperature, and delta G for primer hybridization. And in this model, two random coil strands, A and B, come together to form an AB complex. The temperature at which you have half random coil and half duplex is the melting temperature. And the amount of energy, free energy released in this process is the delta G of the reaction. Now, the two-state model works well if your actual reaction really does involve just these two states, random coil and duplex. However, the problem is that real DNAs are not like this. Real DNAs are folded molecules, as shown on the right-hand side. So your target DNA is a molecule that is often highly folded. And if the folding region, shown in green here, if the region where the primer is going to bind is folded, then before the primer can bind, that region must become unfolded. And that reaction can be slow, kinetically slow, and it also can inhibit the equilibrium of the reaction. So in addition to the target being folded, the primer or probe itself can be folded. And again, before the primer can bind to the target, the primer needs to unfold. And that is also a slow reaction. All right, so at DNA Software, we take this into account, the competition between folding of single strands and hybridization of the two strands in what we call the multi-state coupled equilibrium model. All right, so the multi-state coupled equilibrium model is one of the strengths of our software, and that is important for accounting for false negatives that occur in reactions. All right, this next slide is showing you about some of the causes of false amplification in PCR that can consume primers and cause the PCR reaction for some of the targets to shut down. The first one is widely known. The first mechanism involves primer dimerization, in which the 3' end of each primer hybridizes to one another, and such a hybridization event would lead a polymerase to extend that primer and thereby consume that primer and cause that particular reaction to get shut down that involves that primer. This is a mechanism that is widely understood and accounted for in most software design programs. Less often accounted for is primer cross-reactivity. So the issue here is that in a multiplex PCR, you have many primers and many amplicons, all in the same reaction. And if the amplicon from one reaction, say that you have an amplicon from the Zika virus shown on top here, and below here is a primer from the influenza virus, say you're doing a 2-plex that involves those two viruses. If the influenza primer binds to the Zika amplicon, that cross reaction will consume the influenza primer and create a shorter amplicon, thereby spoiling both reactions for both the influenza and Zika detection. There's also a series of other possible amplification reactions that can occur that I will leave for the users to look at our previous webinar. And lastly, there's the concept, step E here, about off-target background amplification. So if we were trying to make a diagnostic for viruses or bacteria, we would be concerned with the human genome as a possible background. We wouldn't want those primers to amplify somewhere in the human genome. That would deplete the primers, causing also the multiplex PCR to not amplify uniformly and cause problems. I'll return to this in one moment. Let's take a brief aside and talk about the problem of using BLAST to predict primer specificity. Of course, a BLAST search is one of the most widely used methods in bioinformatics. It's meant to determine sequence similarity and to infer evolutionary ancestry, but it's really not appropriate for the use of predicting primer specificity. So let's talk about why that might be the case. So what's wrong with using BLAST to predict cross hybridization? Well, the problem is that BLAST searches based on sequence similarity, not complementarity. So, as a result, users will perform a workaround. And that workaround is to take the complement of their oligonucleotide and then use BLAST to search for hits to the complement of their oligo. And that workaround immediately produces problems. The first problem is that it gives the wrong ranking of hits. So BLAST is searching or ranking the hits based on the amount of similarity. And we'll see why that doesn't give the correct ranking in a moment. A BLAST search misses about 80% of the thermodynamically stable hits because it's using the wrong scoring. It also gives you many irrelevant hits. You know, a BLAST of your primers against the NT database, the nucleotide database, gives you many irrelevant hits that you don't care about. Also, BLAST does not distinguish about hits that are extensible by a polymerase versus hits that are not extensible by a polymerase and not as problematic, therefore. It also doesn't detect amplicons that are formed by pairwise combinations of primers. All right, let's look at why BLAST gives the incorrect rank of hits. And the fundamental problem here is that sequence similarity is not the same thing as thermodynamic stability of a complementary sequence. So for example, not all matches are equal. All right, if you consider a GC base pair is not equal in stability to an AT base pair. So BLAST, on the other hand, because you just take the complement of your sequence and score all matches the same, effectively it is scoring all G matches the same as all A matches, which is equivalent to saying that a GC pair is the same as an AT pair, which is not correct. BLAST also doesn't understand that different mismatches are unequal in stability. It instead is looking at an evolutionary model where a mismatch is an indication of a mutation that's occurred. Very different than what we're looking at with primer design, where we're looking for thermodynamics of complementarity. And so BLAST scores all mismatches to be equally penalized. For example, a GT mismatch, it would score the same as a CC mismatch, when in fact the thermodynamics of GT is a very stabilizing mismatch, and a CC mismatch is the most destabilizing mismatch in thermodynamics of complementarity. It also doesn't account for several other effects, such as the fact that bulges in BLAST are scored based on insertion and deletion frequency. Well, that has nothing to do with hybridization. Bulges are thermodynamically very unfavorable for very different reasons than the reasons scored by BLAST. It also doesn't include dangling ends. BLAST doesn't include the position dependence of mismatches, so mismatches at the end of a helix do not contribute the same as mismatches in the middle of a helix. It also doesn't account for temperature and salt effects. And importantly, it doesn't account for whether it's extensible or not. All right, so now let's talk about an important problem for modern primer design, which is the idea that we can use collections of genomes in our design. So for example, if we were going to design a diagnostic for the Zika virus, we would want to make sure that that diagnostic detected all known variants of the Zika virus. And in this case, we did a search of GenBank and found 168 complete Zika genomes. And certainly we would like any diagnostic we design to detect all of those. We call that the inclusivity list. We'd also like to make sure that our diagnostic does not amplify genomes of near neighbors and give a false positive. So in this case, near neighbors, we would put them into a list called the exclusivity list. And this would be a collection of viruses in this case that are either phylogenetically related to the Zika virus, or are so-called near neighbors, or present similar symptoms in the clinic and hence likely to be in a sample that would give a false positive. And then lastly is a background list where we would want to include any genome of organisms that could cause a false positive. So for example, if we were doing a diagnostic for the Zika virus, well, the Zika virus would be tested from a sample derived from a human being. And we would want to make sure that the human genome would not present a background amplification that would deplete the primers and cause false negatives potentially. All right, well, let's talk about an application of this now. So a common artifact observed in PCR is shown on this slide. This is a dilution series for a target that's been diluted tenfold at each one of these different concentrations. And this is the way PCR should look. It should be that if your target is in high concentration, you get a high amplitude of fluorescence signal. And as you lower the concentration, the amplitude of the PCR signal should stay the same. But in fact, what is often observed is a set of curves like that shown on the right-hand side here, where we see a high fluorescence intensity for high concentrations of target. And then as you get lower and lower concentrations of target, you see that the fluorescence intensity gets decreased. And this should not be happening. And many users are not aware of what the causes of this so-called step-down effect is. Well, it's due to the off-target hybridization to the background DNA that we talked about in the previous slides. So if your primers are, let's say your Zika primers in the previous example, if those primers are binding to human background genomic DNA, well, those primers will get depleted. And if the primers are depleted, then the amplitude of the PCR signal is going to decrease, as shown here. And this becomes more of a problem depending on which is in a higher concentration, the target DNA of interest, Zika virus, or the background DNA. And as you get to lower and lower concentrations of the target DNA, then that background DNA consumes more of the primers and is able to compete in the reaction. So in a single-plex PCR, this step-down effect is somewhat of just a nuisance. But in a multiplex PCR, this effect is one of the major causes of failure of the multiplex PCR. It results in some of the amplicons being amplified at an unequal rate, and that in turn leads to false negatives in the multiplex PCR. All right, lastly is the problem of getting everything in the reaction to play well together. As I mentioned earlier, multiplex PCR is a multidimensional landscape that has a huge number of possibilities of combinations of multiplex interactions. And the iterative empirical approach is suboptimal or fails. That leads to the whack-a-mole I discussed earlier. Even an iterative computational approach will not work. Even though computation, it can try billions of possibilities, billions are nowhere near the 10 to the 30th power combinations that we saw. So multiplex is a complex system, and it requires a 21st century approach. And we have implemented such a 21st century approach into our algorithm. So we call it the multi-pick algorithm, which tests all permutations of singleplex candidates. And it uses a brilliant depth-first search with pruning algorithm to solve that combinatorial explosion in a tractable fashion. And it guarantees that we find the top n best solutions, multiplex solutions, out of the whole space of 10 to the 30th. This is something that is completely different approach than the empirical approach. All right, so we've encapsulated all of the concepts that I've talked about today into our multiplex software called PanelPlex, and we like to think that this makes multiplex design made easy. This takes away the whack-a-mole, and it's encapsulated into our products, the PanelPlex MDX and PanelPlex NGS. And for more information, you can look at the information at the bottom here for contacting us at DNA Software. Thank you for coming to our webinar.
Thanks, John. That was a really great presentation to finish this webinar, and you provided the audience with some really great insight into primer design, as well as showed us the power of PanelPlex. So we thank you for that. Before we start our Q&A session, I want to remind everyone once again that this is their final chance to submit questions for our speakers. So hurry up and send them in now. All right, since it looks like we've got some really great questions that have come in already, let's begin the Q&A and try to get to as many questions as we can. Give us a few moments on our side to get all the logistics squared away, and we'll begin the Q&A presently.
All right, guys, so let's begin the Q&A session. Guys, the first question actually is going to be, I think, one for both of you. You might want to weigh in on this. One of our audience members would like to know how many targets are there in the multiplex? I'll let either one of you answer first, whoever decides is the best person to answer.
John, do you want to go first?
Oh, sure. All right, so the size of multiplex that we can handle depends on which product you're using of ours. So for molecular diagnostics, we did for Arjang's company InSilixa, we did a 32-plex. But we've done much larger plexes in other contexts. So for example, for cancer panels, for targets from the human genome, we've been doing plexes above 50-plex, 100-plex in that neighborhood. And we're currently working on a PanelPlex to extend its capability to above 200-plex. So for most applications for like next-generation sequencing and for molecular diagnostics, the plex levels that we're hitting are the ones that most of our customers are telling us that they need. Arjang?
Yes, so from our platform point of view, generally what limits the level of multiplexing is not the computation and the primer design part of it, is how much product you need to create. In molecular diagnostic assays generally, similar to what we have, in order to do the detection, you need a minimum level of, amplicon generated. In our case, if you want to drive the hybridization and detect them on the array, you need about 10 nanomolar per plex. So if you run the numbers where the capacity of PCR and how much product you can create, you should create, or you can create after 40 or 50 cycles, 40 cycles generally, that ends up being more than 50-plex or, I would say 70-plex would not give you that much targets. That's the limitation of PCR. If you go for the larger panels, as John mentioned, preamp for sequencing and everything, obviously this number can go extremely large. So it depends really on what you want to do. And if you want to do a quantitative analysis or you're using it as a, preamp, you need just 10-fold increase from the background to do the sequencing. Those numbers can be then extremely large.
All right, great. Thanks, guys. Arjang, stick with me. We got another question for you. One of our audience members would like to know what other options did InSilixa consider besides DNA software?
That's a good question. I think we spent about 6 to 9 months. We were looking around for how we're going to do these larger multiplex panels, and we were heavily engineering and bioinformatic biased, so we knew. At least we thought we knew what we were dealing with. We look at all the kinds of wrappers and all the things that you can do around Primer 3 and all of them. We were aware of the models that John had with, in Visual Amp and everything. But at the end of the day, we looked at it and checked the algorithm of John and gave DNA software a lot of pain and suffering. And finally, we decided to go with that. I think that decision was the right one. The results were extremely good. And the rigor was there. So.
All right, thank you, Arjang. John, another question for you. What is the limit of the plex size PanelPlex can design?
That's related to the question we just had. Basically, The NGS version of the software right now can handle up to 100-plex, and we're working to make it go higher than that. The version for MDX, actually, we usually work with customers when we're combining panels, like we did for InSilixa. We do that in a concierge mode, where we will do that in-house to handle the larger panels. And oftentimes, customers have very personalized needs in terms of how those are set up. So they require more hand-holding and more of a sort of consulting mode. But the software itself can handle, quite large plexes, as I said.
All right, thank you, John. Arjang, a question for you. What did you mean when you said that DNA software is the right partner for us? Did you mean to imply that it's not right for everyone?
Yeah, this is sort of my assertion. I would say yes. The reason is you have to have, a level of sophistication, and you want to have a level of sophistication in your primer design, and also have the team that is receptive to it. Because at points it's very much, complex and computationally intensive. So on the concierge mode, yeah, anybody can do it. You can work with DNA software, and they will design and give you the assay, and probably they know more than what you even think. But if you want to actually in-house for a custom method develop it, you just have to have the right bioinformatic tools and expertise and be receptive to it. Sometimes people who are experimental, they rather change a base over here and there to optimize their multiplex, and there's some guidelines that they have, and it just can become really hairy if you do those things and you're not doing it systematically, because you can just get out of hand. The multiplex can get out of hand. You cannot just tweak it manually, if you will. So that's what I meant. That was right for us.
I'll follow up on that. The multiplexing empirical approach, and even with the sort of limited bioinformatics approaches that many companies will take, are often lead to that whack-a-mole situation that I discussed in my part of the webinar, where folks get into this loop of design and redesign and reoptimization, and without knowing what's going on and not really taking into account the system effects. So that's really the essence of what we've spent a lot of time figuring out. All of those causes that make multiplexing not work, wherein like primers cross-hybridize or primers are forming false amplicons with the background DNA. Those issues are not something that typical users have the expertise on hand to make the proper software to be able to do that ahead of time. So unfortunately, many companies that end up coming to us are ones that have spent a lot of time, more than a year trying to get a multiplex to work after great pain. Then they'll come to us and realize that we can help them tremendously and go much, much faster. Just to tell a story, we had one company come to us with a sexually transmitted disease panel that they had been working on for well over a year. They had 2 different groups of scientists work on that, and both sets of scientists were unsuccessful. They came to us with about a month left in the project and said, hey, can you make this 30-plex multiplex STD panel for us? And we were successful and got it to work on the first try. So those kinds of stories happen a lot at DNA software, and a lot of users don't have the awareness that they need in order to realize that they need help, and that DNA software has already invested 15 years of research and development, more than $15 million, making the software that we have, the PanelPlex software, so that they don't have to reproduce, something that's already been discovered, namely PanelPlex.
I just want to add one thing, John, to what you mentioned, and I think is left in the presentation, and that's you also have to know your enzyme and your master mix. Some of the myths that people have that one of them is better than the other one, or I like this kind of enzyme mix because I have been using it in the lab for a long time. That is also something that you need to be careful about it. If you have 3' exonuclease or not, these have tremendous effect on the multiplex when you get over there. And I think that's that's something that also, when you look at the interaction of the primers and everything, you have to be careful about. And sometimes people forget about it and think, you know, they just stick with one that they had, which you have to be careful, right?
Yeah, very often we need to educate our customers about, you know, things, best practices in the field, and that can actually be one of the most valuable services we provide sometimes, is getting people on the right track in terms of, you know, best practices in terms of the enzyme they use, the thermal cycling process that they use, you know, how to properly get the databases for inclusivity, exclusivity, and background. Those sorts of things are not a sort of different way of thinking about the problem, and oftentimes we bring that to the table of really helping people solve their problems.
All right, thank you guys. Very good discussion. John, we have another question for you since you're on the line. One of our audience members would like to know, how long does it take to run a design in PanelPlex?
Depends how big your plex is. If you are, and it depends how big your inclusivity and exclusivity and background panels are. So a typical, like let's say you had, like I mentioned the Zika virus during my talk with 168 targets, and then it had, the background had 5,000 different viruses in the exclusivity, and then we had the human genome in the background. So that panel took around 8 hours to run using our 33 CPUs, dedicated, or 36 CPUs dedicated to their job. So we're doing a massive, massive calculation. You know, these are not something you could run on a laptop computer. So that's one of the advantages of using our cloud-based computing. So each panel, you know, think it should, you know, so it depends on the size. That was a sort of large panel there. Sometimes they don't scale as much as you might think. So some of the larger panels in Arjang's talk that he mentioned had, you know, 7,000 to 10,000 sequences in their inclusivity and exclusivity. Those, we munch right through those. Some of the rate limiting steps are more like how long is the target you're having us analyze, and how many, you know, like, so for example, a virus target is a lot faster to run than a bacterial target, for example. So it can be a complicated answer to that question. The larger plexes that we do for next-gen sequencing, those panels, like a 50-plex, something like that, 50 different targets in the human genome, making a multiplex against that, runs in about 20 hours on our system right now. So that's just sort of two benchmark times.
All right, great. Thank you, John. Arjang, we have a question for you. What's your estimation of efficiency of multiplex strategy for paper-based amplification or lab-on-a-chip operation?
Well, I cannot comment on the paper-based. I don't know exactly what the question is referring to. But on the lab-on-a-chip-based, you know, because at least what we have experience with, generally the transfer that we did from a benchtop instrument to the lab on a chip was pretty successful. The only thing that we had to be very careful about it was obviously any kind of inhibition that happens in a, you know, sort of a lab on a chip platform, you know, and that's generally materials and surfaces, surface to volume ratio. But another thing which was very important was the temperature profile that you have in some of the assays, depending on what kind of structure your applicants have, is very important and has a direct effect on the efficiency. So meaning that, you know, your ramp rates and exact temperatures, especially for the denaturing and all of those things. So if you keep it actually the same as what the instrument is, your benchtop instrument is, which by the way, the benchtop instruments do not have the perfect, you know, sort of uniformity and everything, you know, by far, but, you know, just if you keep it within these regions, I mean, you know, within those realms, I think the transfer and the efficiency would be, you know, similar to what you have in the benchtop. You know, 10%, 15%, different. That's essentially what we have observed.
All righty, thank you, RJ. Folks, with that, we've come to the end of our webinar. So I'd like to remind everyone that the webinar will be archived on our site for about 6 months at www.genengnews.com. So if you missed any parts of it, you can watch it again, or if you know somebody else that might be interested in it, you can feel free to forward the link to your friends and colleagues. I'd like to thank Arjang, Aditya, and John for their informative presentations, and I'd like to thank you, the audience, for your attention and your thoughtful questions. And a very special thanks to DNA Software for sponsoring this webinar. So hopefully we'll see you again at another Gen webinar in the near future. Goodbye for now.